Yeah, I guess we can start. First of all, Professor Sid apologized for not being here to welcome you. So, but in his name, my name, and the name of the physics department, I'd like to thank you, Professor Vlatko Vedral, for accepting the invitation. And as usually we do, I will ask my colleague, Eduardo Olimpio, to do the presentation of today's speaker. Eduardo, please.
So thank you, Fernando. So good afternoon, everyone. It's a great pleasure for me to introduce our speaker today, Professor Vlatko Vedral. Vlatko is a professor of quantum information theory at the University of Oxford, as well as a professor of the Center for Quantum Technology at the National University of Singapore. He is also a fellow of Wolfson College in Oxford. So broadly speaking, Vlatko works on topics that bring out profound implications of quantum mechanics. And for instance, in today's talk, titled Hybrid Quantum Technology, Living Systems Entangled Quantum Bits, we will explore the boundaries of validity of quantum physics. If such boundaries exist, we don't know yet. Maybe he knows if they exist or not. So Vlatko is well known for developing a novel way of quantifying entanglement and apply it to macroscopic physical systems, as well as for characterizing classical and quantum correlations. He's a very productive scientist with hundreds of articles and tens of thousands of citations. Vlatko is also the author of several books, including popular science books such as The Coding Reality, where he vocally indoctrinates people into the idea that information is the fundamental building block of nature. He has also written more specialized books such as Modern Foundation of Quantum Optics and Solid State Quantum Information. Personally, I have had the pleasure of working with him and his group in the last couple of years, where I learned a lot and engaged in exciting projects in a very friendly research environment. I still have this privilege to collaborate with him and his group and still participate in the meetings of his research group. And finally, I have to say that Vlatko has always shown himself to be greatly enthusiastic in visiting us personally. I believe his presence here will be very beneficial for the department. And as soon as we have a more solid improvement of the pandemic, I will try to officially ship him here. So a warm welcome to you, Vlatko. The floor is yours.
Thanks very much, Eduardo. You know that I'm definitely ready to pay a return visit to you as well, and I think that would be a great pleasure to be there as soon as COVID is out of the way. I don't know whether physics will help us with this or not, but I hope it will be over soon. So my topic, if you can see the screen and the slides.
Yes.
Great, excellent. My topic is, as Eduardo said, on hybrid quantum technologies that are at the interface between quantum bits and living systems. But of course, also Eduardo mentioned that the really long-term vision is firmly rooted within physics. So it's a topic that I approach really as a physicist, and I think that's where most of these questions are interesting to us physicists. And really the big question is whether there is a limit to quantum mechanics. Is it really a universal theory, and can you really interfere anything? The honest answer, obviously, is we don't know. There is nothing really to suggest currently that quantum mechanics would fail, which of course begs this second question, which is, let's imagine that there was a limit of that kind. Why can we think, why would there be a limit to interfering, let's say, or superposing more complex objects? And of course, the usual culprits that you hear about are gravity, because we don't really know how to treat gravity properly quantum mechanically. And the other culprit that I want to focus on today is really living systems. And you will hear various arguments there that actually quantum mechanics stops to work somehow when it encounters life, because some people even claim that the definition of life is that it must interact with quantum mechanics in the way to resolve superpositions and ambiguities due to interference. Some people almost credit living systems as the ultimate observers of quantum mechanics. And it may not surprise you that Niels Bohr was already possibly the first person. I may be mistaken, I'm not really a good historian of physics, but he was possibly the first person to speculate on this possibility. He gave a really nice set of lectures called Light and Life. Interesting title in its own right, by the way. This is when they were grappling with the fact that the electromagnetic field needs to be quantized. And the question was still at that time whether and how to quantize light and what would that really mean. And of course, you know, all of this work ultimately led to the, you know, development of QED and so on. But what was interesting is that Bohr, as you know, Bohr saw the principle of complementarity not just at work in quantum mechanics, but he saw it at work in almost anything. Bohr developed a philosophy according to which almost anything in science, and not just physics, but chemistry, biology, even sociology, economics, has to be seen as some kind of tension between complementary properties. And so it's interesting that he postulates, as it were, I guess he guesses in these lectures, that there is a complementarity between the quantum aspects that we see in chemistry, that of course underpins all living systems, and on the other side, organization, which of course characterizes life as well, that there are all of these processes, but somehow they work in a highly organized and synchronized way. It requires a huge amount of coordination and almost design. And Bohr thought, and you can see from maybe the last sentence of this longish exposition, is that Bohr thought that there was a complementarity between chemical basis and organizational hierarchy, in the same way that there is a complementarity between momentum and position of a particle in quantum mechanics. So somehow what he thought is that if you want to study the chemistry, the quantum chemistry, the system will not be able to be alive during that study. So you will either choose a living system, in which case you can study biology and organization, but if you want to study the microscopic processes and properties, you won't be able to maintain life. And what I want to say today is very briefly to completely deny this. In fact, I'm going to contradict it, and I think the conclusion will be that there is almost certainly no complementarity between being alive and being fully quantum mechanical. So that's kind of my view, and you know, to reveal that, I think Eduardo is very familiar with my personal bias, but I think at the moment we really see nothing wrong, either conceptually, either theoretically, or experimentally, with really treating the whole universe quantum mechanically. This may, of course, change, which is why we are going into these more and more complex directions. But at present, I think we don't really have any indications that something will change dramatically. I'm going to tell you in about 10 minutes what would constitute a kind of the ultimate proof that something is really quantum mechanical. And that's what we are after in these living systems. And then I'll describe to you a sequence of experiments that we've been performing and planning as well, which are going in this direction. So the big thing really would be quantum entanglement, as you know, and Schrödinger, of course, called it the characteristic trait of quantum mechanics. And the complexity that we are facing is that if we have many constituents of our system, certainly when you go beyond atomic physics into solid state, into chemistry, into biology, you are facing a larger and larger number of relevant subsystems that you have to look at. And the simple, just speaking very crudely now, what you want to know is whether the overall state of your n quantum subsystems can be written as a convex combination of product states. So these states that we can see in front of us are basically called separable or disentangled, and they are thought to be classical subset of states within all allowed quantum states. So I'm giving a little bit of a reason here that these are states that can be created by local operations and classical correlations. So basically, if you have 2 or more users of quantum information, they never need to exchange any quantum bits. They never need to set up any entangled states between them in order to create a set of states of this kind. And there is a belief, which actually incidentally is still a big open question in quantum computing, whether this a computer that only operates with these states could actually be equivalent to a classical computer. So if you never allow any states of the more general kind than separable states, there is a belief in the community that you would be able to efficiently simulate this with a classical computer. There's no proof of this statement. And it's actually very difficult. It's a difficult question. It's a beautiful question, but it looks very difficult mathematically speaking, which is why, and I'm trying to say this now because the question is, how do we then tell if a given state of a complex system is of this form or not? If we cannot even handle these states classically easily. And in fact, the question that I just raised, given a state of let's say even 100 qubits, how do I know whether it can be written in a separable form? This, in fact, can be shown to be an NP-hard problem in computing. So there is very little hope, at least as far as our current understanding of computation is concerned. There's very little hope that we can ever do this in any efficient way, anything that's less than exponential in the resources as far as memory and time. However, we are lucky that we've been developing witnesses of entanglement, and that's actually all you need to know for this talk, really. So this is a kind of an artist impression of all possible quantum states. I'm drawing it as a convex set, if you like. That's why I chose a circle. And then within this set, we've developed all sorts of other states, which, for instance, are states that are entangled, but we can't do anything with them locally that's of interest. And then, of course, this yellow set is the small subset of these separable states, which, like I said, are most likely not useful to us as far as quantum information is concerned. And the question, now I'm going to ask it purely as a mathematician. The question is, if I'm given a state rho, how do I know whether this state belongs to this red part of the states, red subset, if you like, or the orange subset or the yellow subset? That's as simple as that. And the mathematician would immediately recognize this as a setting for something that they call the Hahn-Banach theorem. It sounds, it's a theorem in functional analysis, and it actually is not very easy to prove. It happens to be a difficult exam question if you're doing undergraduate mathematics. But to a physicist, we are simpletons, and we think of these things in extremely simplified and crude ways, and we don't really care about all the kind of intricacies that may exist in the mathematics and possibly infinite dimensions and all of that. So to a physicist, actually, the answer is very simple. If there is a plane, and I call this plane W for witness. If there is a plane such that rho is on one side of the plane and the yellow subset is on the other side of the plane, then mathematically speaking, you would say that rho cannot belong to the yellow subset. And that's actually all there is to witnessing entanglement. Of course, in quantum mechanics, W happens to be a Hermitian operator. So everything or anything we can measure in quantum mechanics has got to be represented with a Hermitian operator. And the name of the game, therefore, is, can I find a Hermitian operator? I'll write it mathematically now, what it means, really. Can I find a Hermitian operator such that the expected value of this operator with respect to the state rho is below certain bound? This bound could be any number. Usually we normalize it to 0 to make our life easier, while at the same time, all of these yellow states are greater than or equal to that bound. So this is exactly what it means for the state rho to lie on one side of the plane W, and the yellow set of separable states to lie on the other side of this bound. So now the whole ingenuity of what everyone is doing when you are thinking about complex systems and witnessing entanglement in complex systems is actually finding a suitable W that you can really measure in the laboratory. And that's the difficult part, actually. Luckily, there are many natural witnesses that we can think of, and maybe that's what I want to say in a few minutes before I tell you a little bit more about our experiments. This is just a slide to maybe tell you about a couple of places. There are many more places where you can read about correlations, and the story is an extremely beautiful story, maybe that's developed over the last 30 years, where we are now trying to classify and understand the difference between entangled states, separable states, only classically correlated states. And the game is played both spatially as well as temporally. So there's a kind of strong relativistic component to this to also have a unified view of spatial and temporal quantum correlations. Now, for a physicist, what's really important is to understand the following two things, and they are really what makes your life difficult in practice. This is what determines whether your witness is actually relatively easy to access and realistic or not. So the minimum requirements for entanglement are the following two requirements. We need to be able to identify at least two subsystems that we can control individually. We can drive them separately. We know that we can interact with one of them while not disturbing the other one. So that's the condition R1. And the requirement number 2 is that we need to be able to measure at least two complementary observables pertaining to each of these subsystems. So I think those of you that are familiar with Bell's inequalities will recognize this immediately. So basically what you have to measure in let's say 2 qubits, if we are talking about qubits, is you have to be able to measure at least two different non-commuting Pauli, if you like, operators on each of these. And let me give you an example. In fact, exactly what I started to talk about. If you talk about 2 qubits, then a very natural witness of entanglement. And in fact, a lot of the stuff that I'll be talking about experimentally is trying to access a type of correlation of this kind. So this would say, if you can measure XX correlations on the 2 qubits. The X basis is the one where the logical states 0 and 1 are superposed. So the eigenstates of X are 0 plus 1 and 0 minus 1. And ZZ, in my language, or in the language that we now commonly use, is simply the 0, 1 basis. So this one says, if you are able to measure X and Z on each of the qubits individually, and then put them together to calculate the expected value of XX plus ZZ, then this serves as a very natural entanglement witness. So that's very interesting. And here is a very simple way of seeing that. I don't want to bore you with the mathematics, but if the state is separable, and you take the expected value of this quantity, then it can never exceed a unity. So basically, that's because separable states, you'll be taking an average with respect to the qubit 1 times the expected value of x of the qubit 2, and the same for z. So the whole point of being separable is that the expected value of the product of two observables happens to be equal to the product of the expected values. Classically speaking, you would call these events independent, if you like, when the probability for a joint event equals the product of the individual probabilities. And it's exactly this game now that we play, but one level up, if you like. We are trying to discriminate classical probabilities distributions from genuine entangled states. And you can see that a maximally entangled state, one of the Bell states that we all know and like when it comes to, you know, dense coding and quantum teleportation, this state actually achieves a maximum value of 2 for this witness, and therefore, of course, it exceeds the bound by a factor of 2. And you can see that this is very similar to how we talk about Bell's inequalities. And in fact, all of these inequalities, all of these inequalities that come from the field of contextuality, when people talk about measurements that are contextual or non-contextual, they would all invariably boil down to a logic of the kind that I presented here. The punchline now, this last statement that I have, and again, that's what's giving us difficulty in the lab, really, because we don't know if this is feasible. The question really is, can you really measure both X and Z for any system that we choose to experiment on? And that's a big question. Let me give you just an example of two physical, I call them qubits now, but qubits are, of course, not physical systems. You have to actually zoom into a physical system. You have to go into a subspace that you can control well in order to make it into a qubit. But typically, for instance, what you might have is a chain of spins. It's a magnetic, typical description of a magnetic system in the solid state. And they would be coupled through some kind of Heisenberg interaction, XX plus YY plus ZZ. Notice that this looks incredibly like the entanglement witness. So even the Hamiltonian, in this case, is actually a good entanglement witness. So that's why I said the question of whether a system is in a separable state or not is a difficult one in general, and we don't know really how to answer that. However, luckily, in many physical systems, some very natural operators like magnetization, the Hamiltonian, susceptibility, all sorts of macroscopic quantities do actually constitute a good entanglement witness. And I'm giving you another model, maybe more continuous version, basically Hubbard model, where you have a bunch of sites and the particle can hop between these sites. And again, the hopping of the particle is always described as some kind of exchange interaction, XX plus YY. So this is the interaction that destroys a particle at one site and creates it at the neighboring site, and vice versa. So this, of course, physically is now due to tunneling, not due to the Pauli exclusion principle, but it boils down to more or less the same Hamiltonian, in fact, and this itself is also a good entanglement witness. But the question for us really is can you really now, given this Hamiltonian, can you independently measure X and Z? And I'm not going to really, this in its own right is a very beautiful and important question, and there is much literature on this subject actually. So I don't really want to go into that here, but there are sometimes rules which we call super selection rules, which might prohibit certain superpositions and measurements, therefore, of certain observables from taking place. There is a big question mark there that I put, and this is due to the fact that we are still debating whether superselections are fundamentally there, or they are simply due to various environmental influences that may have to be controlled better in order for us to be able to overcome these super selection rules. So a famous prohibition in quantum mechanics, for instance, is the one that does not allow you to superpose even an odd number of charges, of fermions in general. In fact, I always have electrons in mind. So for instance, a state where you have 1 electron plus 2 electrons is a state that, according to some super selection, should not actually be possible to prepare or to measure even in this basis in the lab. If this is the case, then of course we already have a problem with witnessing entanglement in systems like that. So I'm just mentioning this to you as an important subtlety, although again, in what I will be saying now, this probably will not play any role. But because I motivated the whole thing with limitations to quantum mechanics, is there any limit to what we can superpose and what we can entangle? I thought it would be worth mentioning super selection rules. So let me tell you now a little bit about experiments. This was an experiment that was done by Dave Coles a few years back. And we were really motivated by this statement of Niels Bohr. The question was really, could we have a system that's living and at the same time quantum entangled with something? And as it happens, the easiest thing to do in this case was, in fact, to take one of these purple bacteria that are actually much investigated in the field of quantum biology, because there are all sorts of claims that these bacteria use quantum effects for photosynthesis. And the idea was really to try to confine a single bacterium between two. These are mirrors in a microcavity that are very close to one another. And basically, the question is, by coupling this bacterium very strongly to the light that's present inside this cavity, could we then create an entangled state between the bacterium and light while the bacterium was actually alive? So these are experiments that we've been very lucky actually to have a private individual who some 10 years ago, Jim Martin, 10 years ago decided to make a huge donation to Oxford. I think he gave Oxford University £200 million. It's the biggest donation. And we were doubly lucky actually that he identified us as doing something interesting, I think. And well, he didn't give us all 200 million, but he gave us 1% of that actually, which I thought was a good investment. And in fact, these experiments were really experiments that were made possible, among many other things within this research, because of Jim Martin's interest. And I'm actually a great believer of funding of research in this way. I think governments are failing us a great deal. They tend to fund very incremental research that frequently is not very exciting. And I think most of what I'm telling you about is actually privately funded these days. And like I said, we are lucky that there are some crazy people out there who think that this actually is an interesting topic. So let me show you some images from this. Maybe more interesting to show you now, more speaking as a physicist, to show you energy levels within this bacterium. So what the bacterium does is it has an antenna. These are, by the way, bacteria that live about 1,000 meters below the sea level. And they receive they receive a handful of photons per day, actually. So that explains to you why they have evolved to be extremely good at catching these photons and converting them into energy, into chemical energy, ultimately, as Bohr would say. And this, of course, is paramount to their survival under those conditions. So they are masters of photosynthesis, if you like. They absorb light by a device that resembles very much an antenna, almost like a satellite dish that's evolved to capture this photon, which then trickles down. It loses energy, if you like. You can see that the wavelength is increasing from 750 nanometers, just to give you roughly an idea what these transitions are. Ultimately, as it loses its energy in a radiationless way, this is basically lost to vibrations inside the organism, it reaches this reaction center where it runs an ATP cycle to generate energy. And the rest, as people would say, is really chemistry. But the interesting bit to a quantum physicist, I'm sure there is something interesting even in the ATP cycle, but the real interesting bit that we wanted to focus on are these energy levels here. And those are the energies that we wanted to match to the frequency of light inside the cavity. And that's what Dave Coles really did. So here are the images. So what we wanted to show is that these energy levels are shifted due to the coupling with the electromagnetic field in the cavity. This is something that quantum opticians would call vacuum Rabi splitting. This is a genuine quantum mechanical effect that comes when you treat light in a quantized way and couple it in the dipole electric field approximation to another system, material system. In this case, this happens to be a living bacterium. And actually, what Dave and I spend a long time discussing is how do we tell, so we can tell whether the bacteria are entangled to light, and I will show you those images in a second. But the question for us was really, how do we know that they are alive while this is happening? We really wanted to show that the system could be living and entangled at the same time. Much as a response to the challenge that Niels Bohr actually put forward to all of us. And actually what we did is, what Dave ultimately came up with is something that biologists use very frequently, is he introduced a dye molecule, which you can see here, fluorescing in purple, if you like. You can even see it. So in this case, the dye molecule is outside of the bacterium. And this is basically how you tend to tell very crudely as a biologist that a system is in fact living, because it's keeping any foreign material outside of itself. Homeostasis, if you like, is I think the word that biologists, it maintains its own integrity. However, you can actually show that when the bacterium dies, then the cell membrane simply no longer presents a challenge to outside molecules, and you would actually be able to see a dye molecule penetrate the bacterium, and then you can see it fluorescing from within. So it's a very crude, I know that this is a very crude witness of being alive, but I think it's the one that's experimentally maybe the easiest to access. Of course, we did a lot of quantum modeling. I don't want to go into the details of this really, but the Hamiltonians and techniques that we would traditionally use in the solid state, much like the Hubbard model. In this case, I called it the Holstein model. It's one version of that, basically transporting in solid state systems while being coupled to vibrational degrees of freedom. And even a simple model like this, it's extremely hard to apply to a biological system, simply because things like T, which is the hopping rate between sites, which you need to describe how this energy propagates between different sites, and the coupling to phonons in my language here, in fact, very difficult to get at experimentally speaking. So frequently you have to do all sorts of numerics in conjunction with experiments in order to get some idea about these relevant parameters. And again, I may recommend this, one of our papers here, if you really want to read about this in more detail, but I don't really want to bore you too much with these details. Now, here is a slide from Dave's experiments on a single bacterium being coupled to light. So he literally had, maybe I'll go back to show you that, he literally had a white light, not a laser driven. This is a completely incoherent lamp, white lamp light, literally shone through the cavity, exciting the bacterium, and then waiting for the leaky cavity to emit radiation and detect it. And that's what the pictures here are all about. And what you can see literally is that as you are changing the length of the cavity, you are coming closer and closer to being on resonance with some of these levels that exist inside the bacterium, in which case you can see this famous signature. If the bacterium was not coupled to light, these two levels would simply become degenerate and they would cross one another. But because of this vacuum Rabi splitting, as I called it, because of the entanglement between the bacterium and the quantized radiation, these levels actually tend to avoid one another. And you can see this presented at different frequencies, each of which actually corresponds to a different transition that you can tune into, focus into, zoom into, if you like, in this experiment. So that's our witness, if you like. It's a very crude witness of the state where the dipoles inside the bacterium, which you can treat as very crudely again, as a harmonic oscillator. So each of these dipoles you can think of maybe as a qubit, but even a single bacterium has hundreds of thousands of these dipoles that will simultaneously all couple to the quantized electromagnetic field. So at a very simple level, and that's the model that I want to show you. So here are these, I call them living polaritons, because here you can see basically this is a shot of the bacterium inside the cavity, then also showing that the dye molecule is outside of the bacterium, which presumably means the bacterium is still alive. And at the same time, you're getting spectroscopy to tell you that in fact a vacuum Rabi splitting is there and it's observed during this experiment. So we went in actually with theory. This is work that I've done with Chiara Marletto, Dave Coles, Tristan Farrow, who I think is also present here. In fact, he's been developing most of these experiments in the lab, and I will tell you about some of them as well in a few minutes' time. We went in with a very, as you would expect a simpleton physicist to do. We went with two harmonic oscillators, one representing the bacterium and the other one representing the quantized mode of the electromagnetic field in the cavity. Then we coupled them together, dipole-dipole interaction. And in fact, the entanglement that we are really talking about is the entanglement that exists in the ground state of this system, where you have some amplitudes, Cn, to have n photons in the cavity and capital N minus this n. So basically, the photons exist either inside the mode of the cavity of the quantized electromagnetic field, or they have excited the dipoles inside the bacterium. And in quantum information, there has been a huge amount of work in understanding these kind of many-body entanglements. So it was very easy to actually apply the existing theory for this. One entanglement witness, in case you're wondering, that we were able to relate to is simply dispersion in the positions of these two harmonic oscillators, and at the same time, the dispersion in the momenta. When the two are maximally entangled, this witness here goes down to 0. In fact, it's the lowest. That would be what we call the EPR entanglement, the maximum entanglement between two harmonic oscillators. However, we couldn't achieve that. I was more in the limit where lambda, the coupling between the two, is smaller than omega. And we actually, I think, managed to get to roughly 10 or 20% of omega, lambda being equal to that. That's how strong the coupling was between the two. So we could detect a certain amount of entanglement. We are deviating from the classical bound, but we are certainly not violating it as much as one could violate maximally. So this would be one picture where really this coupling strength matches the frequency of these harmonic oscillators, in which case your entanglement really becomes almost like what you would call an order parameter in condensed matter. It shows you that there is a phase transition from a kind of loosely, slightly entangled system to a very entangled system. I think, like I said, our experiments are more towards the beginning of this onset of criticality, maybe 20% of entanglement that we managed to achieve. We were certainly not able to probe the region where we are maximally entangled. So there's a lot of work on these kind of things. Now, what we wanted to do if we had time, and I think this is something that we are planning to do now, and I will show you another experiment in a different context where we ended up doing something similar, is not just to observe the Rabi splitting inside this living bacterium, but maybe to add a coherent laser light that could drive transitions between these split energy levels. This is again something that we call Rabi flopping. That would, of course, go further in the direction of showing that we can treat this hybrid system between a bacterium and light as a genuine quantum bit of information, that we could indeed prepare any superposed state and so on. More importantly than that, what we would like to be able to do in order to conclusively talk about entanglement, and I said this is our biggest challenge really, and I will show you how we intend to overcome that. What we would really like to be able to do is perform separate measurements on the two subsystems. In this case, this is really impossible because the bacterium is sitting right in the middle of the cavity, and it's very hard to perform measurements on the light in the cavity separately from the bacterium. They come together as dress states, and those are the measurements that we have, and those were the easy, relatively easy measurements to make. But actually what we would really like to do to be much more conclusive is to be able to address the two systems independently. And then, of course, what I mentioned earlier, the requirement number 2, that we are able to access two complementary observables is really the main obstacle. That's something we have to figure out how to do before we can do these experiments. I might mention, before I tell you about the latest experiment, I might mention that Dave had all sorts of other, you can see that we are going in the direction of Schrodinger's cat, because if you detune the levels of the bacterium sufficiently, you will be able to make the bacterium completely incapable of photosynthesis. So the bacterium is tuned at a very particular frequency or wavelength of light, and if you strongly couple it to the cavity, this frequency will ultimately shift enough that the bacterium will no longer be able to photosynthesize. So I call that quantum bactericide. You know, killing bacteria is probably something we would like to be able to do with viruses, as you know, as well these days. But this is kind of interesting because it begs the question of, could we actually do this in a superposition to, in fact, create a living and dead state of the bacterium. And of course, this would be stunning. This would be the ultimate in all of these experiments. And as you know, all experiments in quantum mechanics are kind of versions of Schrodinger's cat experiment. So is it really possible even to go in this direction? And how does one do that? And how is this scaled up and so on? But the last thing I really want to tell you about is what I mentioned to Eduardo, actually, and he thought this would be of interest. These are the latest experiments that I've been working on with my colleagues in Singapore. I think the person here who is really in charge of these experiments is Rainer Dumke. And on the theory side, Tomek Paterek is the person who actually suggested most of these ideas. So this is a completely different qubit. This is no longer quantized light. This is actually a superconducting qubit. And because superconducting qubits exist at very low temperatures, the challenge here was to find an animal, in fact, a living system that can survive millikelvin temperatures. So these are temperatures that are 1,000 times colder than the room temperature, 10,000 times actually colder than the room temperature. And to our great surprise, there is an animal that's called a tardigrade, a slow stepper. It's a tiny animal. Actually, you certainly need to amplify it. You need a microscope to see it. You can see the size roughly there. It's 1/10 of a millimeter. Some of them can be slightly even larger to 1.5 millimeter. And what we decided is to actually couple this tardigrade, as you can see it in this picture, to one of those superconducting quantum bits. And in fact, what we wanted to see is exactly the shifts in the energy levels, much as we did in the previous experiment. We wanted to see the shifts in the energy levels that would indicate to us that this animal here is actually coupled to the superconducting qubit. And instead of doing just coherent transitions on this coupled system, what Rainer managed to do actually is add another superconducting qubit and entangle it to this first hybrid system. So we kind of took it a little bit further than the previous experiment, because now you have two quantum bits, if you like. One of them is a hybrid living system and a superconducting qubit, and the other one is just a superconducting qubit. And Rainer basically showed that you can prepare all sorts of entangled states of this kind. So individual local Rabi flopping as well as entangled states between the two. This paper is just finished actually, and I think will be on archive soon as well. I'm happy to send you a copy if you're interested in reading about it. Here are some movies just for fun, if you can see them. The motion of these tardigrades are remarkable animals. They were sent on a rocket to outer space. They were actually subject to the to the cosmic microwave background temperatures, very cold, and they very low pressures as well. And in fact, when they returned to Earth on a rocket from one of these experiments, to scientists' surprise, there was more tardigrades present than there were on departure. And in fact, so not only did they survive couple of Kelvin temperatures, but they actually managed to multiply under those conditions. They are remarkable animals. They have the ability to go into a state of TUN, T-U-N, or a state of complete suspended animation, in which actually they can get rid of the water from their system. And this is the one that's sitting exactly at the Josephson junction, if you like, inside the capacitor, and changing the energy levels of this qubit while Rainer and his group are doing the experiment. So this creature is remarkable because what happened is, in fact, like I said, is that during this experiment, which lasted a long time, we're talking about days long exposures during this experiment, the tardigrade actually managed to survive millikelvin. So not even Kelvin, it went down 1,000 times lower. And I think we're talking about pressures that are a microbar. We are talking about pressures that are about 1 billion times lower than the atmospheric pressure, million times lower than the atmospheric pressure that we feel. So both temperatures and pressures were extraordinary. And of course, the system tardigrade went into this state of suspended animation. And after the data were collected, Rainer put the tardigrade back into a droplet of water, and the guy completely revived and went around. Usually they just go around looking for food. I think they're very simple animals. They eat continuously. I don't think they do much else, actually. I don't think we understand them well. We don't even understand how they have these amazing survival strategy. Maybe there is something there for environmental scientists to really learn about. But for our purposes, I think the fun was really to show that this tardigrade can, in fact, entangle to a quantum bit. Now, I didn't want to show you too many pictures from these experiments because, again, what you will invariably see is what I'm going to say on this slide, is that when you look at the resonances of a superconducting qubit without the tardigrade being present there, they are modified when you couple the tardigrade, and you will see the energy shifts, much as I showed you the energy shift in the case of Cole's experiment with bacteria. It's just that now we have a much more complex organism biologically, and it seems to be a perfect organism to actually try to do with these kind of cryogenic temperatures. Now, the best explanation I can give of the kind of state that the tardigrade reaches with a superconducting bit is very much akin to how we explain the Casimir effect or the van der Waals force. So this is a genuine effect of the quantum vacuum, if you like. It's an indirect proof, these van der Waals forces, that that quantum vacuum in quantum physics has to be ascribed a degree of reality. It really can affect things. So here again, I have two coupled harmonic oscillators. You can treat each of these very crudely again. You can treat the superconducting bit as a harmonic oscillator. You can treat the tardigrade or whatever couples inside the tardigrade to this, whatever dipoles there are, as a harmonic oscillator collectively. And then you have the usual dipole-dipole interaction. And when you diagonalize this system, I think London, these are sometimes referred to as London forces. I think London was the person who actually wrote the paper in Nature in the 30s to explain this. When you diagonalize this system, you will see two frequencies. So these are your energy shifts from the original energies that characterize these two harmonic oscillators individually. And you can see that the new ground state, if you like, the 0 point energy of this system is in fact lower than the 0 point energy when the systems are not coupled to one another. And it's this lower energy that we attribute to the van der Waals forces, as you know. So in this sense, again, very crudely and very loosely speaking, a tardigrade forms a kind of a complex molecule with a superconducting bit. And quantum mechanically, the explanation lies in the fact that the energy of coupling is actually negative, and it gives you an energy that's lower than the energy you could reach if the two systems were not coupled to one another. The reason why I say that classical physics cannot explain this, of course, is that the energy, as far as classical physics, the lowest energy is always zero. And that's because momenta and positions can simultaneously have the value equal to zero. So the whole Hamiltonian, the whole energy, classically speaking, could be equal to 0. But because quantum mechanically, because of the uncertainty principle, this is of course not possible. That exactly is what leads us to this kind of van der Waals or Casimir energy. And in fact, everything in quantum electrodynamics, one might say, is just a consequence of these kind of vacuum fluctuations. You can phrase the whole quantum electrodynamics in this way. So this is kind of the best explanation that I can offer at this stage. Let me tell you what we are doing now, and this is something that Tristan Farrow is working on. Actually, here is a proposal that luckily I'm saying here. So Jim Martin was the person who gave us the initial funding to do the experiment with bacteria. What we now propose really to do is to double this experiment, to take 2 cavities, to basically trap a bacterium in each of these cavities, a living system. I'm saying bacterium because probably if you go with this technology, this will be the easiest thing. A virus or bacterium will be the easiest thing to handle. And then simply have a source of a single photon that you split at a beam splitter. So this is exactly Schrodinger's cat experiment, where the photon is in a superposed state of going one way and the other way simultaneously. Then it gets absorbed either by one bacterium or the other bacterium, in which case what you're creating is an entangled state, excited ground plus ground excited. So that's what we wanted to do. And like I said, we are very lucky that there are foundations out there. In this case, this is the Moore Foundation, that's actually given us a comparable amount of money, about 2-3 million pounds actually, over the next 5 years, in order to try to achieve this kind of entanglement. So this would really be an entanglement where we are aiming at preparing 2 living systems. That they must be really living during this experiment. I think that's really the challenge of this. But we want the 2 systems to be in a genuinely quantum entangled state, where we can even violate Bell's inequalities with this kind of system. Once you separate them in this way, there is hope that you can, in principle, measure each of them individually and then somehow conclude that they are really in an entangled state. So this is something that we are trying to do. And then, of course, you can think about arrays of these systems and so on. The main motivation, like I said, is really scaling entanglement up. There is no indication that these hybrid systems would make better qubits than what people are developing now. So we are not really thinking about them technologically speaking. The question really is just a fundamental question. And so let me leave you with these conclusions. I'm gonna not read out these lines. I'm gonna leave them for you to read. What I'm gonna say is the following. It seems to me, so there are many interesting questions here, physics-wise or otherwise. In fact, biologists, whenever you talk to biologists, they immediately say, oh, is this kind of quantum state useful for biological purposes? So biologists may not care about quantum mechanics, but they do care about the biological side. Is there an evolutionary advantage to these systems really utilizing quantum mechanics? Is it really deliberate? Has nature, if you like, has natural selection pushed systems towards actually really implementing some simple quantum technologies? I think that's a great question for a biologist. But what I want to conclude with is how I started. I motivated this by quantum mechanics, leaving the micro domain of atomic physics and going into the more and more complex domain of chemistry and biology. And I think this seems to me like a hugely exciting direction of science. I think this will define physics going and leaving the boundaries of physics and going into chemistry and biology will define the next 50 years of science, in my view. I think that there will be a great convergence of our understanding of nature at that level. But one thing I want to say is the following. It seems to me when people study quantum mechanics at that level, what you most likely are to read is speculations where quantum mechanics fails. So people will say, oh, maybe at some point entanglement vanishes, maybe we will not be able to violate Bell's inequalities and so on. To a person like me, it seems that these questions are wrong, actually, based on what we know now. I would be more inclined to ask the question the other way around. Is nature going to give us correlations that are even stronger than quantum correlations? So I doubt that we are ever going to go back to classical physics. It's almost certainly inconsistent to have a half of the universe quantum and half of the other half classical. And actually, the papers with Chiara that I'm citing here are very much along the lines of ruling out all sorts of hybrid systems. My bet is more that we should be testing whether the correlations that we are ultimately detecting in some other domains even exceed what quantum mechanics allows us to have. And to me, it seems that this is more likely to lead us to something novel than some kind of return to semi-classical or classical reality. Thank you very much for your attention.
Great. Let's open the microphone to thank the speaker. Wonderful lecture. So, any question, comment, please raise your hand. We have Fernando José, please.
Hi, Vlatko. Great to see you after some nearly 20 years.
Hi,
as
long
you were
time.
visiting Waterloo with Elham.
Yes.
Anyway, as you know, I'm a mathematician, so I'm trying to make sense of a bunch of things. One of them is that you invoked Hamiltonians that is non-constructive. Yet, Hamiltonian doesn't need fully the axiom of choice or several others hard to find in physics things. It needs actually that you have a sort of probability, and that's the case in every physical quantum system that you see. So probably the presence of Hamiltonian is still allowable and compatible between the non-deterministic side and the actual physical realization. Yet, I'm thinking about that Hofst's advocating that there is only one physical system, that is the universe. And as you discussed, specific aspects of the living beings being entangled, how much of the living of the being is actually dependent on this entanglement? In other words, why would not this be like those macroscopic oscillators that already are entangled, like these experiments that made this macroscopic sort of Schrodinger's cat? So actually, the question seems to be what depends on life, or what life depends on this particular sort of control. That is what we do in quantum information, right? We use control, or we use systems with very localized energy levels to simulate isolation, insulation.
Yes.
So
Yes.
I think that the liveness, the life of the living being, actually may not be playing a fundamental role, and the living being is acting as an inanimated being, essentially. Thank you.
It's a great question, actually, at many levels. It's a good question because let me convert it into the following one. If you were to repeat the bacterium experiment with a dead bacterium, you would actually see probably very similar energy shifts. So in that sense, what you are detecting is probably not crucial for life at all at this level. In fact, what you would have to do is what I was suggesting on one of the slides, that you might want to displace the energy level so much through strong coupling that you may inhibit this ability to do photosynthesis. But of course, this does not at all prove that there is anything quantum mechanical in this sense, because you can do these shifts in a classical way, as you say. Let me make a more general comment than that, because I think your question is important precisely because of this. Whenever I go to a conference on quantum biology, actually, this topic gets hotly debated, because the question really is, what would be a non-trivial effect in biology as opposed to something that's given trivially to you. So, for instance, we all know that the existence of atoms would be impossible, without quantum mechanics. The whole of chemistry of molecules and all the molecular forces are ultimately genuinely quantum mechanical. If you switch quantum mechanics off, you wouldn't get molecules either. You wouldn't get solids, as you know, and nothing else really. There would be no matter. That, in a way, would be considered trivial, if you see what I mean. The whole structure of the system would just be given, even though it would ultimately be quantum mechanical. So in that sense, trivially, quantum mechanics, of course, is important, but I don't think that that's the kind of quantumness that we are after. I think what we would like to really show is that you could get entangled states even at the level of complexity or timescales, which would actually be surprising. You could get coherence that would last longer, for instance, than you would expect from some kind of naive decoherence model that you can apply. This would already be extremely exciting to a physicist like me. But I think you are completely correct in saying that even this would not actually prove anything about the need of this kind of stuff for life. And I think that itself is a big question, but I don't really know how to begin to address that. I think what you would have to engage in is probably some kind of genetic modification of the bacteria in the way that you change the structures of these complexes, so that, for instance, you really show that the constituent parts are so separated now after the mutation that, in fact, there couldn't be any coherent quantum transport in this system anymore. Maybe at that point you show that also the system can no longer function. So it would be interesting. What I'm suggesting is to genetically probe things that at the same time tune in and out the system from being able to utilize quantum mechanics at some level, and then really show whether this is or isn't detrimental to its state of being alive, if you like. I think these would be great experiments, but no one has done anything like that so far. And like all I can say is that I hope there are more crazy people out there who would like to invest into research like this. I think it's very, very exciting.
And I think that when you talk about the scaling issue, you have to consider the different omics, right? Because in life, you definitely have omics as regimes for scales. So
yes,
definitely they will be playing a different role at each of these regimes.
yes, you're absolutely right. I mean, maybe to even say something concretely, when you look at this photosynthetic system, you are usually talking about the noise comes from phonons, which are usually very high frequency. So we are talking about phonons that are between probably, I'm going to say it in the inverse manner. if you look at how coherent they allow the system to be, is usually picosecond, even smaller time scales. And so what experimentalists then decide to do is really probe the quantum features beyond these timescales. So they're thinking, if decoherence is likely to destroy quantum transport on picosecond timescales, let's see what happens on nanosecond timescales, 1,000 times longer. And what surprised many researchers in photosynthesis is that they still saw quantum mechanical oscillations which persisted in this system. And then all sorts of theories kind of came out of that. How does the system do it? Is there some kind of error correction and so on? So there's an awful lot of kind of beautiful ideas going in that direction. But I would say all of them are inconclusive. They are wide open. We don't have evidence either way, really.
And it's also the time scale is important for these different omics. So in spite of maintaining the coherence, you may not be able to see the full interaction if you don't consider the scale for a life event. But
yes,
thank you so much. Thank you so much, and great to see you.
Thank you.
Fernando, let's give a chance to our colleagues to
Eu acabei de
do some questions.
eu acabei de dizer tchau.
Okay, Eduardo, I guess you raise your hand, right?
Yes, yes. You talk about stronger correlations in nature,
Yes.
like than those in quantum mechanics. I have two questions. So the first one is, do you refer to life properties or conscious properties when you talk about that? Or in the other question is, when I see this experiment involving living beings, I always think of conscious beings participating in this experiment. Not the Wegener-Strand paradox, but conscious experience.
It is a great question. I think it's a great question, Eduardo. Yes.
And then what if the tardigrade could speak after you open the box and you talk to him? So
yes. I love your question. Actually, I'm hoping that in parallel to this living system, you are completely right. When you compare a bacterium to a cat, then of course it's much more boring in this sense. In fact, even a cat is boring compared to a human being. You would ultimately like to do Schrodinger's cat with a human being, simply because it's another conscious entity that you can communicate with faithfully, right? What I'm hoping, unless we learn something really surprising about the brain, I think working with a human being is probably too challenging, unless there are bits that we don't really understand there. I mean, there are many bits we don't understand. But what I'm more likely to suggest now is to use a quantum computer to simulate artificial intelligence. That's an interesting question. So what if we could, for instance, with 1,000 qubits or million qubits, it doesn't matter. What if we could really simulate universal artificial intelligence? This would be a completely closed system, completely coherently developing. And then I think it would be very interesting to do what you're suggesting, to in fact try to create two orthogonal states in a superposition with this system, and then probe its internal states. How does this system perceive this? What does it feel? Again, you probably can guess what my bias is. I think a closed quantum system is perfectly capable of being conscious. I'm kind of a strong AI supporter, I would say, in this respect. Again, based on what we've seen so far, I'm not saying there won't be some huge surprises there, but I think what you're suggesting actually would be extremely interesting to try to probe. Let's make an artificial intelligence system, and let's try to really make it in a superposition of different states, and then probe its internal dynamics. What does it mean to be in two different states of perception if you like.
It's a very good idea. I haven't thought about that. But the other question, you're talking about stronger correlations.
Yes.
Can you be more specific about that than those?
Yes, I think I usually I'm usually thinking that this is more to do with not to do with biological systems necessarily, because they are very messy. They are not excluded, but I'm more thinking along the lines of theories that will supersede quantum mechanics. For instance, when we talk about quantum gravity, it is very possible that the theory that comes is different to quantum mechanics and gravity, if you like. It somehow, in special cases, it reduces to the two. But when you put them together, you really get a theory which goes beyond what quantum mechanics allows. So somehow, I think our intuition should be suggesting to us that the kind of the theory we should be looking for, it's probably going to do better than quantum mechanics, if you see what I mean. It's unlikely to be worse as far as quantum entanglement is concerned. It's more likely that it can do more than quantum entanglement, and only under special circumstances it will actually reproduce quantum entanglement. So I'm kind of thinking that whatever comes after quantum mechanics, and we don't know what it is, if there is anything, it seems to me it's going to be in the direction of stronger correlations.
Great, thank you. Thank you, Vlatko.
Any other question? Fernando Paris, please. Go ahead. I guess your microphone is not working. Oh, man. Oh.
Not yet.
Nope. I guess you have a...
You can write in the chat.
Yeah, I'm looking also at the messages. I can probably read the message if you type it up.
Yeah. I guess he's writing down, so. Okay, a comment about the R2 requirement.
Very good. Do you want to say more? Oh, I see. Are you referring to the fact that you could measure only 3 instead of 4 observables? That on one subsystem you can measure 2 complementary observables, and on the other one you only measure 1, as in the original Bell inequality. I'm not sure if this is what you... No, maybe that's not what you have in mind.
No. What a shame.
I could comment something on that, just in case that's the intuition. I think it really depends on the extra assumptions that you make. In some cases, you can indeed dispense with the R2 assumption, but it usually implies that you've made certain other assumptions about the collective quantum behavior of your system. Whereas if you really want to make as few assumptions as possible, then somehow it seems to me this R2 assumption is very difficult to avoid in some sense. And of course, it's the one that we would love to be able to avoid, because our experiments would be much easier frequently to do if we didn't have to demonstrate both of these.
Okay. Maybe you should move you on to another question.
Yes.
If Fernando can fix that.
We cannot hear you. Paris, we cannot hear you yet.
Yeah.
Yes.
Okay. Any other question? Someone else would like to comment? So, okay,
I have one more.
Okay, Eduardo, please.
These tardigrades, I suppose they are very massive. Yes, of course.
They are very massive. You're right. They are much bigger than the bacterium. I think our assumption was to take this 1/10 of a millimeter size, so like a cube, and in a way, it leads you to estimate maybe some kind of either dielectric constant that you want to use to model it as a dielectric or as a bunch of dipoles and things like that. Yes, it's actually very massive, really.
And then I wonder if the superposition of these tardigrades cannot generate a detectable superposition of gravitational fields. I'm changing the subject, but you are saying that you tardigrades in superposition, and then maybe you can. Of course, you are changing in a superposition.
That's a beautiful question, Eduardo. It was suggested to me by an experimental physicist in Vienna who actually thought that these ideas that Chiara and I had on testing the quantumness of the gravitational field using two massive superpositions, maybe that could be combined with two living systems being entangled through gravity. I think that's a great idea. That would be an impressive experiment if you could do that. The difficulty I'm imagining is you would now need to superpose the tardigrade in the center of mass. So, what we are discussing here are really internal superpositions of different energy levels. But in order to make it maybe gravitationally relevant, you should really try to make a spatial superposition. And this seems to me extremely difficult, really, how to do.
Yes, because when you have a superposition of internal degrees of freedom, you still have this superposition of geometries, right? But at the same point, and I suppose that because it's in the same point, it's difficult to detect that, right?
That's it. That's it.
In different points, it's better to see the movement around the
Yes. Yes.
Yes, I see. Yes, perfect. Thank you.
I think this, please, proceed.
Yeah. Pedro Maciel asked a question on the board, and Fernando Paris as well. So maybe you can check that.
Let me see. Thank you. Is there any work on applying quantum algebras in quantum biology? Good question. I have not seen any work in that direction. None of this. Actually, this could be interesting as well, since we are searching for maybe a way of expressing this quantumness differently. We know that, in physics, we have a bunch of ways of thinking about this algebraically, geometrically, logically. In fact, quantum logic is there to tell us about interference and superposition and entanglement. I have not seen anything on Hopf algebras and quantum groups in quantum biology. That was the first question. And then the second one is this R2 thing that I have to have a look at. That's a PRL from 2019 on quantum non-locality in a triangle network. So this is 3 parties that actually can make a single measurement and dispense with R3. That would be great. I'll have a look at that. That would be great. It might be easier than to go with 3 subsystems, as you say, providing that we can only perform 1 measurement on each of them.
Wonderful. Any further question?
Thanks.
Okay, if you have no more questions, thanks again the speaker. Thank you very much. It was a wonderful lecture.
Thank you.
Okay, goodbye then.
Goodbye. Take care.
Bye. Thank you. Thank you very much.
Bye, brother. Thanks a lot.
Bye-bye.
Bye.